Hello everyone, my name is Sergio Antonio Gomez Lopez, and today I will be taking to you about selection and characterization of Burkholderia for their plant growth promoting effects and influence on maize seed germination. Okay, a Burkholderia in extremely versatile bacterial genus in the environment and has been reported to have great potential to promote plant growth through different mechanisms. This study evaluated plant growth promotion mechanisms in 26 Burkholderia strains. The capacity of the strains to promote plant growth was evaluated through production in indole 3 acetic acid EAA under different pH conditions, substrate, and present or advance of tryptophan, exopolysaccharides production, and quorum sensing. Okay, introduction. The plants provide a wide variety of niche for growth and proliferation for a great diversity of microorganisms, including bacteria, fungi, protists, nematodes, and virus, which make up the plant microbiota. These microorganisms come from complex interaction with plants and have key roles in promoting plants' productivity and health. The plant-bacteria interaction has been the basic of the production that prompts increased productivity of agricultural crops essential inoculants with mechanisms that promote plant growth, nutrient acquisition, and increases of plant tolerance and resistance to abiotic stress and pest attack. Okay, materials and methods. Bacterial isolators. We evaluate total number of 26 per bacterial strains belonging to the microorganisms collection from the Microbial Genetics and Biotechnology Laboratory Academy unit of Garanus and Federal Rural University of Pernambuco, Brazil. Bacteria strains were isolated from sugarcane plants cultivated at the Carpina experimental station. Uh, excuse me. Um, indole triacetic acid EAA production, the EAA production was evaluated via a colorimetric method based on observance in UV spectrophotometer and described by Cruiser et al. The 26 bacterial strain were tested for their EAA production potential in the presence and advance of tryptophan under different conditions on substrate and isolation. The samples were evaluated in a spectrophotometer with the absorbance at 530, and a positive result was confirmed via formation of a pink color. Okay, exopolysaccharides production. In order to detect EPS production, the bacterial culture was transferred to sterile discs on the surface or agar medium containing 10% sucre and pH 7.5 and the temperature of 28 centigrade for 48 hours. At the end, the production of EPS was detected visually via the production of aloe, whose present characterized the result as positive and quantified according to the halo's site. To confirm EPS production, the bacteria positive for EPS production were transferred to tubes containing 2 uh, milliliters of ethanol with a platinum lobe. That test was considered positive when the precipitation of EPS could be detected and negative when the tube remained cloudy. Okay, quorum sensing molecular production. The identification of bacterial strain produced of quorum sensing molecule was performed using a biosynthene bacterium, also called ELH, Agrobacterium tumefaciens NTL4. The tests were prepared in via where EI tumefaciens was inoculated uh, in a line at the border of a petri dish containing uh, Luterina bertani, medium weight 10 microliters of X-gal 8. The bacterial strains were inoculated perpendicular to the line of Atumefacies MTL4. The discoloration at the blue color of the tumefaciens colonies after 48 hours of inoculation indicates the ALH production with the selected bacterium strain. Okay, a genetic diversity of Burkholderia is sp. The genetic diversity of Burkholderia isolates was verified by the repetitive sequence based or PCR technique for the RFP-PCR was prepared in identifying fraction contained the primers box 1R, Eric 1, and Eric 2. Separability for each bacterium strain, the amplificent fraction were done in the the Mocky Claire. White thermal conditioning 95, excuse me, 95 Celsius for 2 minutes. Okay, the second PCR reaction used the primer Eric 1 and Eric 2 was performed to a final volume of 25 microliters containing 0.5 to 10 mg of DNA template, 100 microliters of H primer, 2.5 ml of H DMTP 10x PCR buffer, 50 ml of magnesium chloride, and 5U microliters of Taq DNA polymerase hot viral pulp. The amplification reaction were performed in the thermoclear program with thermal condition of 94 Celsius for 5 minutes. The PCR production were checked via gel electrophoresis, and his DNA segments were visualized under ultraviolet light, and photo document the bands observed were used to build in a similarity dendrogram via Jacquard, confident using passwords. So, okay, a Maize sets inoculation test. A Maize sets were used to evaluate the effect of 11 bacterial strains in improving germination attributes to perform set inoculation. The pure culture of bacterial strain were diluted in PBS buffer to an optical density of 1 at 630 mn. The mice sets were immersed into suspension for 30 minutes under constant shaking. Inoculate sets were planted in Germates paper were water rolled up and stored at the temperature of 28 Celsius with a photo period of 12 hours for 7 days. The experiment consisted of a completely random scene. This thing with a control and 10 trends with 3 uh, replicates. We sampled 725 sets per replicate. Okay, in this table, we can see control on 11 bacterials inoculate applied on the Maize sets in the germination test. Okay, statistical analysis. The normativity of EAA, FPS, and corn set grown promoting data was checked with the champion wheel test. Then the data were submitted to ANOVA and the significant interactions were tested with the Scott-Connell test at 5% probability level using the past software. All data complete were evaluated in the bioplot of principal components analysis based on the correlation matrix B1. The observation and the evaluated variables under study. The multivariable analysis was performed using two kind of the person correlation matrix were uh, plotted in the air program. Okay, results and discussion. The PCA view plan comprised 75.41% of total data variability in bacterial strains in AA production with or white old tryptophan addition, the presence of sodium chloride, and under different pH values, the first principal component explaining 58.88%, and the second explaining 70.53% of the total data variables. Variants. Okay, in this picture, we see by pilot for two principal components based on the correlation matrix between a bacterium from the genus Burkholderia and the production of EAA in different tryptophan and sodium chloride concentration. Okay, tryptophan plays an important role in the EAA biosynthesis, and its presence in the culture medium increases the phytohormone production by the majority of TGPV in the first quadrant, the bacterial strains Bedouin, Gladioli, and Bedouin B20, Burkholderia spp, were positive correlated with the presence of tryptophan in the medium, producing 226.7 and 1 and 250.54 micrograms of IEA emulate respectively and significantly differing from the other bacterial strain. Okay, oh, excuse me. Producing of indolacetic acid in vitro in the advance or press of tryptophan pH values 4.5 and 7.2, and sodium chloride concentration bibacterial lineage belonging to the genus Burkholderia. And conclusion, the results showed that the genus Burkholderia is capable of synthesizing EAA under different abiotic conditions, specifically the B10 bacterium Burkholderia ambifaria stood of producing a great amount of EAA, regardless of the advance or presence of tryptophan pH or concentration of sodium chloride in the culture medium. Most of the bacterial studied shown a positive response for production of EPS and quorum sensing molecules. Straining of Burkholderia spp also shown a high potential for colonization, bacterialization during seed germination, promoting increased in-row length, short length, and total fresh and dry biomass of the corn celery. Thank you for attention. This is a reference